Microphone better. No microphone? Okay, thank you. Okay, so thanks for being here. I'm Bernardo. I'm from the American Museum of Natural History, but I'm originally from Brazil, so I'm very happy to be in this meeting here. And I'm going to talk about a little bit about the parasitic wasps I study and how to study these convergent adaptations, or at least what seem to be convergent adaptations. So basically, the very nice looking wasps that you see here are the critters that I study, the cryptine wasps. As you can see, they're very nice looking. They have these very diverse color patterns. They're generally very small and extremely diverse. There's about 2,400 species of them all over the world, from polar regions to the tropics, to deserts, and so on. And they're parasitoids, right? They're parasitic wasps, which mean only the immature states have this parasitic lifestyle. The adults are free living and they go after their host where they lay their eggs, the eggs develop as parasites, and so on, right? Basically, they feed upon a wide variety of hosts. Beetles, moths, other wasps that make nests, and so on. And these hosts are usually concealed to various degrees. They're hidden. They're usually pupae, they're hidden somewhere. Many other parasitic wasps, for example, the attack hosts are exposed, right? Like this unfortunate caterpillars here. But cryptine wasps, in contrast, they usually attack hosts that are hidden. For example, here under a leaf row, or sometimes in twigs or vines, they pupate hidden inside these places exactly so that they can avoid predators and parasites. And in some cases, the hosts are really, really well hidden under very hard substrates, such as these tree logs here. So this wasp is actually drilling with the ovipositor inside the tree log to find a host that's hidden within, inside, right? This is a tree log, and some other wasps attack other wasps that are making nests out of clay. They get the mud, they make a nest out of it, and it hardens a lot. It's a very hard clay, and so they have to drill inside it and so on. And to accomplish that, to attack these deeply concealed hosts, they have to, many groups have evolved interesting features that seem to be ecomorphological adaptations to find and assess these hosts. So, for example, many of them have the tip of the antenna modified, and in order to sort of hammer the substrate. So they go around with the tip of the antenna and they tap the substrate, producing pulses of sound. Those pulses of sound are then detected by those reception hearing organs here housed in the front leg. So here's sometimes the segment of the leg where these organs are is quite enlarged. You can see here the leg is quite swollen in comparison to like another random cryptine, right? So they have to develop these hearing organs really well to find the echo, so basically to echolocate their hosts, right? Once you find a host, you need then to, of course, drill inside the substrate. And for that you need very strong oviposition muscles. So here you can see a contrast in the tip of the abdomen of this wasp here, very enlarged muscles here, versus these guys here, right? So this is another putative adaptations to access these deeply concealed hosts. And also you need a thick, stout ovipositor, right? In contrast with these like very slender ones, right? So all of these seems to be adaptations to find these deeply concealed hosts. And finally, many groups, they developed a lobe in the ventral part of the ovipositor that sort of hugs the dorsal part, so that you have a firmer grip when you're drilling into hard substrates. And at last, if you're going to spend your life developing inside of wood or other hard substrate, once you reach the adult stage, you need to chew your way out of the wood, right? So you need strong mandibular muscles. You can see here a stark contrast between this bulky guy here and this slender guy over here, right? So all of these adaptations, they make cryptine wasps really interesting to study the evolution of traits depending on different parasitic lifestyles. But the problem is that cryptine wasps are often considered a difficult group. So when I go around and I say to people I study cryptine wasps, this is the kind of thing I get. Anyway, I would be glad to meet a guy crazy enough to work with cryptine. We currently don't have anyone who deals with our cryptines. It's very hard to distinguish cryptines and so on, right? Because basically a lot of people seem to think they're difficult. And that stems basically from their sheer diversity, right? There's so many groups, so many genera, so many species. it's hard to even start tackling them as a group, right, as a systematist. And also they present this confusing array of similarities and differences, which means a lot of character conflict. So you cannot really find good characters that split them into diagnosable groups. So when you do like a morphological phylogeny, you find high rates of homoplasy. It's really hard to classify them anyway. So classification is hard, their classification is in disarray, and we don't have much of a good phylogeny on cryptine wasps. The most valiant effort so far was made a few years ago by Lorraine and others. But you can see here, it was the phylogeny was based on a single locus, and you can see here it's quite unresolved, mostly because of problems with indels, right? So right now we don't really know a lot about the evolutionary history of cryptines, which makes it hard to answer interesting questions about their these adaptations, like how many times did these characters evolve? Are they correlated to each other, or do they evolve independently? You know, you can have like strong oviposition muscles without really having a good echolocation system, or you need to have one to develop the other. Or if these characters are evolving in convergent fashion, how structurally similar are them? How similar are the muscles that evolve in one wasp, in one lineage of wasp, as compared to the ones that evolve in another one? So to address this question, the first thing we need is a good phylogeny, right? So basically I spent the last 3 years of my life trying to build a robust phylogeny for cryptine wasps. I went all over the world to collections and museums and to field sites trying to assemble a good taxonomic sampling. So basically, as for now, I have a little bit more than 200 species from 170 genera, which means about 70% of the genera from pretty much all over the world, as you can see here. So I basically got this, I sequenced 2 mitochondrial markers and 5 nuclear markers for all these wasps. There's a bit of missing data because you don't get good PCRs for everything, because you go to museums and you have to get things that are sitting 70% FNO and so on. But it's not like a terrible amount of missing data, so I think it's robust. And then I analyzed it with a variety of methods. For the sake of simplicity, I'm going to show you the likelihood results today. And then I did ancestral state reconstructions using my maximum likelihood and MK1 model. Basically to try to address how many times these characters evolve, and if all these wasps that display these adaptations are in a single clade or what, right? And then I tried to correlate the characters among each other and with the use of deeply concealed hosts themselves. For that I used the Pagel method, which basically is based on a likelihood ratio test that compares a model in which these characters evolve in a correlated fashion versus a model where they evolve independently from each other. So I don't want to torture you with the details of the classification of cryptines, but I think it's worth showing that, you know, the current classification that needs to be improved, right? These are the color codes here show the subtribes in which the tribe is actually divided right now, and you can see all of them are scattered pretty much everywhere in the tree, right? These are 4 of them, but there's 15. So basically, most of them are scattered everywhere in the tree. There's only a handful of them which seem to be monophyletic. So definitely someone needs to come and address this problem, try to come up with a better classification that makes a little bit more sense than this mess. And then showing you a little bit about the evolution of these characters. Basically, spoiler alert, all of them evolve like really in a convergent fashion. So they evolve many times in the tree. So modified antennal tips evolved 6 independent times. You can see here color-coded. Swollen 4 tibia, 13 independent times. Enlarged abdominal apex with specialized muscles, 7 times. And the lobe in the ventral valve and the ovipositor evolved a whopping 17 times. So, and finally the strong mandible muscles evolved 8 independent times. So basically all these characters are appearing everywhere. And usually, as you can sort of see in the tree, they don't get reversed, right? It's like they evolve once and they keep going that way. You can see from the trees here, in the trees you can sort of think, oh, the bulk of the diversity for these characters are in this group here, and these are just like, you know, like small things happening like here and there. But to put things into context here, remember this is a very diverse group, so these pair species here, it's a genus with 100 species. Right, so, I mean, there's lots of species with these adaptations, and they're scattered everywhere in the pre-time tree of life. And then finally, I did the results here for the Pagels correlated method. You can see like p-values are microscopic, so there's a strong, very strong correlation between these characters and the use of deeply concealed hosts, right? And also these characters are strongly correlated to each other. Like all the p-values are tiny, right? If you correlate modified antenna with swollen tibia, modified antenna with an enlarged abdomen, and so on, all of these characters are correlated to each other. These p-values are so small, I kind of didn't believe it. I said, oh, you know, there's something wrong with my analysis. So I went ahead and just checked for other random characters, morphological characters, and tried to correlate them with these, and I didn't get significant results. So I think it's kind of believable now. So basically, to start answering those questions that I posed before, how many times did these characters evolve? Many times. Any way you put it, if you reanalyze those data with parsimony, with Bayesian inference, you're going to get the same result many times. And yes, I'm pretty confident to say these characters evolved in a correlated fashion. So it's fair enough to call them a complex morphofunctional system that is specialized to attack, to find and assess deeply concealed hosts. So basically, the next step that what I want to do is to expand the number of taxa and loci. I already have more stuff sequenced and so on. To ask that question, how these structures, they evolved several times, but are they really the same thing? So it'd be really interesting to do functional morphology, ultrastructure, you know, go hard into electronic microscopy and so on. And in the future also address the convergence of lifestyles in terms of morphospace, right? Instead of these key adaptations and so on. Maybe it would be easier to just ask, do these convergent lifestyles, do they impose a constraint in the morphospace that's occupied by those images, right? So with that, I'd like to thank funding sources, NSFD did, the Society of Systematic Biologists, a lot of curators that helped me, like, generously letting me borrow their material, to receiving me in their institutions. I'd like to thank Sylvia Pavan, who helped me with the analysis, and all the team at the Richard Gilder Graduate School at the American Museum at large, and so on. That's it.